Beta-lactamase enzymes hydrolyze bacteria, rendering standard antibiotics useless against bacterial infections. This mechanism, part of the growing crisis of antimicrobial resistance, or AMR, leads directly to increased morbidity and mortality worldwide. The World Health Organization identifies priority pathogens, notably the ESKAPE group: Enterococcus faecium, Staphylococcus aureus, Klebsiella pneumonia, Acinetobacter baumannii, Pseudomonas aeruginosa, and Enterobacter species. These exhibit high levels of multidrug resistance. For example, methicillin-resistant Staphylococcus aureus, or MRSA, modifies its drug target site, altering PBP2A to evade beta-lactam drugs. Current projections estimate that without intervention, AMR could cause 10 million deaths annually by 2050. Phage therapy offers a potential alternative mechanism of action, specifically designed to bypass these established bacterial resistance pathways. Bacteriophages, commonly called phages, are viruses that exclusively infect bacteria, representing the most abundant biological entities on Earth. A typical phage structure includes an icosahedral pamphid head containing the nucleic acid genome, a contractile tail sheath, and specialized tail fibers. Phage infection relies on high specificity, requiring the precise binding of these tail fibers to specific bacterial surface receptors. These receptors are often lipopolysaccharides or outer membrane proteins unique to the host. This narrow host range ensures that phages target only pathogenic bacteria, while preserving the beneficial human microbiome, a key advantage over broad-spectrum antibiotics. Phages are fundamentally classified based on their replication strategy. They are either lytic, meaning destructive, or temperate, meaning capable of integration. Lytic phages are obligately virulent and are the primary candidates for therapeutic use because of their immediate destructive action. The lytic cycle initiates with absorption, where the phage irreversibly binds to the specific bacterial cell wall receptor. Penetration follows, involving the contraction of the tail sheath, which injects the phage genome directly into the host cytoplasm. The phage immediately hijacks the host machinery to replicate its own genetic material and synthesize necessary structural proteins. Late-stage gene expression produces two key enzymes, Hollands and endolysins. Hollands create pores in the inner membrane, allowing endolysins to access and degrade the peptidoglycan layer. This degradation causes osmotic lysis, effectively bursting the cell and releasing hundreds of progeny virions ready to infect neighboring bacteria. Temperate phages can enter the lysogenic cycle, integrating their genome, known as a prophage, into the host chromosome. This integration is mediated by the enzyme integrase via site-specific recombination. The prophage replicates passively with the bacterial cell division, remaining dormant until environmental stress triggers induction. Induction leads to excision and subsequent entry into the lytic cycle. Temperate phages pose a significant safety risk in therapy because they can mediate generalized or specialized transduction. This process transfers bacterial genes, including antibiotic resistance or virulence factors, between different hosts. Therefore, therapeutic phage candidates must be rigorously screened using whole genome sequencing to confirm obligate lytic behavior and the absolute absence of lysogeny-associated genes or toxin-encoding sequences. Monotherapy using a single phage strain risks the rapid evolution of phage-resistant bacteria, typically through mutations in the receptor binding site. To mitigate this evolutionary pressure, phage therapy universally employs a cocktail, which is a mixture of two or more distinct lytic phages targeting the same bacterial species. These phages must utilize different receptor binding mechanisms or possess distinct host ranges to ensure comprehensive coverage and prevent simultaneous resistance development. Cocktail design is based on the specific bacterial isolate from the patient, requiring rapid diagnostic identification and susceptibility testing, known as phage typing. The preparation process involves large-scale propagation, followed by stringent purification to remove bacterial endotoxins, specifically lipopolysaccharides, and final formulation for stability and clinical administration. Phage administration routes are precisely tailored to the infection site, including intravenous injection for systemic sepsis, topical application for skin and soft tissue infections, or aerosol inhalation for pulmonary infections such as cystic fibrosis exacerbations. Unlike traditional antibiotics, phages exhibit self-dosing pharmacokinetics. The phage population replicates exponentially at the site of infection as long as susceptible bacteria are present. This localized amplification allows for significantly lower initial doses compared to traditional drugs. Phage clearance is primarily mediated by the reticuloendothelial system, particularly involving the liver and spleen. The half-life varies depending on the route and formulation, typically ranging from minutes to a few hours in the bloodstream. Phage therapy offers two critical advantages over conventional antibiotics. First, the high specificity minimizes dysbiosis, preserving the commensal microbiota essential for host health and colonization resistance. Second, phages demonstrate superior efficacy against bacterial biofilms. Biofilms are complex matrices of extracellular polymeric substances, or EPS, which shield embedded bacteria from both antibiotics and immune cells. Many therapeutic phages encode specific depolymerase enzymes, often located on the tail spikes. These enzymes specifically degrade the EPS components, facilitating phage penetration and access to the embedded bacterial cells. This enzymatic action makes phages highly effective in treating chronic infections associated with indwelling medical devices or persistent wounds. The primary challenges for widespread phage adoption involve regulatory standardization and the host immune response. Phages are currently regulated as biological products, but their dynamic, replicating nature complicates traditional pharmaceutical approval pathways requiring fixed dosing. In the United States and Europe, treatment often occurs under compassionate use or named patient exceptions for life-threatening infections. The host immune system recognizes phages as foreign particles, leading to the production of neutralizing antibodies which can reduce phage efficacy upon repeated dosing. Furthermore, the presence of bacterial debris and endotoxins in phage preparations necessitates stringent purification protocols to prevent adverse inflammatory reactions in the patient. Synthetic biology techniques are fundamentally transforming phage therapy by allowing precise genetic modification of the phage genome. Engineered phages can be designed to broaden their host range or, conversely, to increase specificity against a particular bacterial strain. One advanced strategy involves loading phages with CRISPR-Cas systems specifically designed to target and degrade antibiotic resistance genes, such as NDM1 or KPC, directly within the bacterial cell. This approach effectively resensitizes the pathogen to existing antibiotics. Other modifications include incorporating genes that enhance biofilm degradation or preventing the phage from integrating into the host genome, thereby ensuring obligate lytic behavior and maximizing therapeutic safety and efficacy.